The silence is broken by somebody crying, trying to be heard, never a word. Always the attitude, sort out your own, always alone, wishing for something the world is denying. Out in the wilderness somebody's crying. Somebody wishing for something to happen, wishing to tell, wishing to help. Someone was listening, someone who cared.
Hello, my name is Ayana Young, and I welcome you to Unlearn and Rewild, where we explore radical ideas relating to Earth renewal. Today, the conversation with Radical Mycology's Peter McCOY probes onward as we invoke the powers of fungi in earth healing and the integration of human societies into our delicate and compromised biosphere. We begin with cutting-edge uses of fungi for personalized medicine, which promises sweeping advances in healthcare and natural healing. We surmise the potentials and the limitations of myco-remediation in confronting industrial agriculture, logging, oceanic plastics, and other major pollution. We'll also discuss the imperative to preserve fungal diversity and the methods of creating local spore and culture banks. Despite being sidelined by academia and the social mainstream, understanding mycology and fungal ecology and the universal patterns therein is an essential tool for reining in the ecological crisis. Moving on to the health benefits of mushrooms, which I know is a topic of great interest to a lot of people, and even more frequently now in the Western world, so it seems. Even though the health benefits of fungi have been well known, as you've mentioned, in Asia for thousands of years, particularly for their anti-cancer and immunity-boosting qualities. My philosophy is eat fungi daily, which doesn't just mean mushrooms, but krauts, miso, kombucha, ciders, yogurts, kefirs, cheeses, etc. And then there are the more fringy uses, such as microdosing with poisonous mushrooms, such as the jack-o'-lantern mushroom, which supposedly causes cancer cells to self-destruct, called apoptosis. So could you say something about the lesser-known or more cutting-edge ways fungi can benefit our health, including a very exciting application being developed by a few mycologists, which is personalized antibiotics?
Yeah, absolutely. So, as you mentioned, fungi are the grand chemists of nature. They do so much for plants and plant roots, and all the fertilizers we give plants is just a poor human reproduction of what fungi naturally create through their incredible chemical mastery. So the same can be said for medicines. Probably a lot of the medicines we attribute to plants is actually a product of the fungi that live inside of the plants. And that might be hard to hear for some herbalists out there, but the increasing amount of studies are proving that 100% of all plants are permeated with fungi, that trillions of different species can be inside, or at least colonies can be inside of one plant. Every plant is a unique quilt of fungi, if you will. And all those fungi are producing compounds, a lot of them medicinal. So that has huge implications for even our understanding of natural medicine, our history of medicine. I think the history of health is really a history of the fungal queendom. And so today we're starting to acknowledge that slowly a bit more. And some of the easiest medicines to make and the most controllable are those made by mushrooms, the more macro fungi that are easier to interact with. But that's not to say that, there are desert fungi from the Sonoran sands that produce anti-cancer compounds. So we don't have to look at just mushrooms. And they produce compounds, mostly sugars, complex sugars that boost our immune system. 90% of their cell walls is comprised of complex sugars, many of which are medicinal. So the fungal tissue is vastly medicine in this way, depending on the species. And then they also produce other ones that have anti-inflammatory and soothing effects. So, and then another thing along with that is getting what you were getting at is as fungi grow, and sort of going back to what I said earlier about the autonomy of every hyphal tip, is as a hypha encounters, say, a virus or fungus or bacteria in its environment, it will sort of war off with that thing and sort of go into a little battle mode. And if everything works out and it has time and the resources, it will figure out the correct antibiotic or antifungal compound to produce to fight off that organism. So that's how fungi survive everywhere. That's how they don't get killed by MRSA, because they're constantly adapting to all the things that are attacking them. So we can take advantage of that as humans. And we see this all the time in the cultivation lab, where you grow fungus sterilely on a petri dish. Some sort of other mold gets in there by accident. The mushroom you're growing encounters it. A couple days or weeks later, after warring off and sort of figuring out what to do, it'll start to just grow right over that thing. Basically figured out what the thing to produce to kill it and wins the battle. So we can do this with perhaps our own diseases. If we have, again, MRSA or some sort of antibiotic-resistant microbe, theoretically we could swab that out of our wounds or mouth or whatever, put it in a petri dish, let a fungus encounter it. The fungus will produce the correct compound, and then we can extract, concentrate, and prescribe those compounds. So it's really custom myco-medicines, because even the flu, right, it can mutate in so many ways. That's where you get a different, I don't get the shots, but you're supposed to every year because the thing's mutating. And if we work with the fungi, it's going to be a much more natural compound. Of course, the fungus can have negative compounds that can produce toxins. Fungi do this. So I've said in the past that in theory, and certainly the practice is there, we can do what I just described, introduce the fungus to the competitor, and then continually introduce the fungus to that competitor so it keeps producing that compound, and then extract that compound. We can do this in a liquid form. So it's a very simple method to grow mycelium in basically sugar water, and we could even introduce this contaminant. But at the end of the growth, that liquid will probably be full of this ideal antibiotic, but it might be full of other nasty stuff too. So the challenge is we don't want to just straight drink that liquid, because it's going to have unknown number of other things in there. Really, that's when it gets to more, the chemistry of it, the synthesis, or not the synthesis, but the concentration and extraction. Some chemistry, I know it's a scary concept for people, and it's not my forte, but it's not as complex as we often, again, just the science is always presented in this inaccessible way, and chemistry doesn't have to be presented that way. It is a challenge for everybody. But I think that what I'm speaking to is that my hope is in the future, as we refine these protocols, which are just beginning to even be looked at or even thought about, hopefully that we can develop methods for doing what I just described, customizing our medicine and then extracting it safely and producing it at home for almost next to nothing, and perhaps even curing largely incurable diseases. The last thing I'll say as far as cutting edge goes, so that's a huge concept, huge, but we're not there yet. It's sort of the unfortunate takeaway for now. I would say another huge topic that I address in the book that my friend James Weiser developed, and he's had some pretty profound anecdotal stories that he's told me, nothing documented yet or as far as peer review, but if you're going to believe the stories like I do, it's pretty significant. And that's essentially that he's growing medicinal mushroom mycelium on grains, which is a normal cultivation practice, and you can do it very cheaply at home in the kitchen, no lab, using these liquid methods I just briefly sort of touched on. But long story short, he's growing the mycelium on grains and then juicing those grains, basically making a raw fungal extract. Most mushrooms, or even medicinal mushroom stuff, we have to use alcohol, we have to use heat, something to break down the cell wall to extract the compounds, which of course is going to mess with the medicine and all the enzymes and all the other good juju in there. So he's just creating a pure juice, pretty simple concept, and getting it all right in there. Doesn't taste super great, but it's not horrible. And he's working especially with lion's mane mushroom, which is one of the more neurological mushrooms. It really helps stimulate your nerve growth, can clear up the plaques in the brain associated with Alzheimer's, and help rebalance your hormone levels or your neurotransmitter levels. And he's told me incredible anecdotes of people with lupus, with fibromyalgia, with Parkinson's disease, really significant disabilities. Just after a few days of drinking this stuff, they're able to open jar lids. They're able to stand for the first time in 5 years. And I'm not exaggerating. So I do mention this protocol in the book, but I do caution, there's always risks with new techniques, but this one, it seems so significant, so simple. I definitely encourage more people looking into it.
Wow. Fungi are so generous in aiding our well-being, and I feel really called to reciprocate and support their communities, either directly or indirectly, in my work in forest restoration. So a question I'm curious about is the resilience of fungi in this age of ecological instability. After heavy logging, the canopy structures of a forest takes hundreds of years to return to a functional state. And I'm wondering, do fungi become established on much shorter timescales, or do many species need the old growth dynamics? How delicate are those ecological webs we've been talking about amid excessive human disturbance? And what is a reasonable goal for restoration?
Well, fungi, again, going kind of back to the balancing or the paradoxes they present, is on one hand, they're so profound, so significant, so unparalleled, and all these fantastic, amazing things. But at the end of the day, they are another delicate organism. They're like everything else. They have nutritional needs. They'll die above a certain temperature. They need water. So we need to nurture them just like they nurture us when we work with them. So that's a big component of understanding good cultivation techniques, is just understanding the fungal needs and reflecting those. And a lot of it just comes down to basic biology and ecology, just where and how do they grow naturally, and we're trying to reproduce that. And the ecological webs that they produce as a part of their work take millennia to produce. Some of these, old growth forests take hundreds of years to get to climax state. Fungi are integral to that entire process, as I've sort of touched on. And when that gets clear-cutted and compacted and churned, all those connections are lost. The mycelial webs are devastated. And it can take decades at the very least for them to start to stabilize a little bit more. There's one study that I read, and I cited in the book, where I think it took about 41 years in a forest for different waves of fungi to sort of stabilize as plants were getting more and more established. So different fungi have different roles. In decomposition, you'll have successional waves of different things eating different stuff at different times. Same things when the rebirth and the regeneration is occurring. You have different fungi coming in. In a bare rock, lava flow landscape, lichens are the first things to show up. Lichens are 95% mycelium. That mycelium demineralizes the rock. Powerful acids release the minerals, providing the first basic soils for plant seeds and everything else to come. And eventually you get the forest, all thanks to the fungi at every step of the way. When we're working in reforestation practices or regeneration practices, we always want to have that fungal component in the forefront of our minds, really, because they are nature's ecologists, they're their stewards. We can never restore the environment. We can never go back to what it was, right? And no habitat even on its own. If you sort of clear cut two forests that look quite similar, they'll never recover the same way. Again, it goes back to the chaos. Everything has its own way of developing, but we can guide that, but we'll never go back to what it was. So this is, I think, one of the reasons why in some places we have really low fungal diversity. I think we've just lost a lot of the fungi because we've destroyed the earth so much, and they are so delicate and their spores just aren't there. So we can intentionally bring in species. Perhaps, again, of course, we want to stay relatively bioregional at the most, and hopefully as most local as possible when we're trying to work with fungal species and diversity. We can inoculate those wood chips like you talked about in the slash piles, and we can do all kinds of just casual inoculations when we're hiking, and even in a relatively protected forest. There's always human impact, and we can help encourage fungal growth in subtle ways. When we're doing anything with soil, we always want to bring in the soil fungi. There are soil fungi that don't produce mushrooms. These are called the arbuscular mycorrhizae or endomycorrhizae, and they are considered the most ecologically significant of all the fungi, which is a pretty bold statement. And that's because they have a significant influence on the structure, basically the fluff or tilth, porosity of soil, which is important for oxygen and water flow, for everything else in the soil. They do a lot of the chemical mastery that I mentioned in the soil, so they're huge down there. And then all that work directly influences the entire assembly of the plants in that entire ecosystem. So all the above ground ecology is influenced by these fungi. And of course, the animals depend on the plants. So at the core of all this is these non-mushroom forming, sort of cryptic, underground, hidden fungi that are everywhere. They associate with 95% of all plants. They're found everywhere except Antarctica. They're some of the most globally distributed and, again, most significant fungi, but most people never hear about them, don't think about them because they don't form mushrooms. You don't see them. Well, you can easily cultivate them. You can easily bring them back into a disturbed habitat. There's relatively simple protocols that I outline in the book that, a little learning curve you hop over, and then you can do it pretty simply to bring in, again, these locally adapted species, locally adapted cultures that are place-based and self-designed, I guess, to regenerate that habitat. There are commercial products. You can buy these things, but I've seen studies where those commercial products, even though it's the same species, can actually have a detrimental impact as what might be naturally present, because again, going back to the idea that the mycelium does adapt to its immediate needs. So you have these local subspecies or strains or cultivars, whatever you want to call them. And those are the most place-based. Those are the ones we want to work with. And again, we can harvest some of those spores with very little impact to the environment, boost them up through cultivation, and then reapply them. And then there's many other practices throughout the book where we can bring in lichens. Lichens are completely overlooked by pretty much any ecological assessment. Lichens are a huge component to nutrient input, especially nitrogen. They fix 50% of the nitrogen in a forest is provided by decomposing lichens. And we can cultivate them. We can even bring them into desert environments where the desert is disturbed. The desert soil, we can stabilize that with desert soil lichens. Practices like this are in the book. So, but again, there's some points I get to like a broken record where I just keep saying there's this important thing. There's very little research about it or very few people looking into it. So, I strongly encourage more people messing around with it and let me know what you discover, because we just need more people on the team. And that's why I've always said that radical mycology has to be a grassroots process, because just the funding and resources and all the higher top-down approach isn't working, and it isn't even really there.
Somebody wishing for something to happen, wishing to tell, wishing to help. Someone was listening, someone who cared, never despaired. Someone to lean on and someone to trust, who needs your assistance and finds your disgust. Something that you have the power to give or to take, to make or to break. The innocent spirit of somebody's hope, assistance to cope. The water that heals or the fire that burns. Out in the wilderness, silent returns.
To ask about a specific project you are working on with the mycorrhizae at the last radical mycology convergence on a Midwest farm that had been abused using pesticides, chemical fertilizers, etc. So I'm wondering if you could explain this experiment and how it could be scaled up to myco-remediate large industrial monocropped lands.
Well, so that experiment was more of a demonstration for the landowner. She does a lot of ecological work, has a lot of people come to her land to visit. And so that one was, again, more of a teaching tool. And what it was briefly was we dug out several plots in this really just old, disturbed ex-cornfield on her property that had just seen the trials and tribulations of industrial corn farming. And it was just covered in really hardy, really, really tenacious weeds. Cleared that out, and equal-sized plots. In some of the beds we inoculated, we didn't do anything. We just put some cover crop seeds on. Other beds we put compost tea and the seeds. Some we put mycorrhizal fungi in the seeds. Some we did biochar in the seeds, and then the other ones we did combinations of all those things. And I've been actually trying to get her to write up a full update about that. But she said that she can definitely see the difference with the mycorrhizae, which is not too surprising, because there actually is, despite everything I've just been saying, or kind of the last thing I said about being overlooked, the arbuscular mycorrhizae, these soil fungi, are the most studied, perhaps, of all these fungi, next to the mushrooms, especially as far as plant relationships go. Arbuscular mycorrhizae are their relationship with plants is the most studied symbiosis in the plant world, hands down. So we do know a lot about them academically, but most people day-to-day don't know about them. So it wasn't too surprising that these fungi helped out the plants. What I think is more interesting is what we're going to be doing at this year's convergence, which we have yet to announce, but is going to be announced quite shortly at radicalmycologyconvergence.com. And we're planning out the installations there, one of which is likely going to be taking the soil from the nearby woodlot, where it's been less disturbed for the last few decades, You take that soil, you screen it through some simple sieves, get all the dirt out of the way, and at the bottom sieve, you're left with the spores of these arbuscular mycorrhizae. You clean out the spores, you separate them. Under a scope, you can identify them and count them. So it's a little bit boring, but that way you can get a baseline about what their diversity is and their abundance per, say, like kilogram of soil. Take those spores, which we're trying to do pretty soon, grow them over the season, over the next few months, with quick-growing plants like grasses, and they will associate with the grass roots in a pot. And at the end of the season, you'll have this soil filled with roots that are infused with mycelium, the mycelium itself, and also spores from the fungus. And all that is viable inoculum of this local place-based culture. And then we're going to take that and put it back into the land and re-inoculate the new plants. And they're doing a lot of restoration efforts and regeneration efforts. And then they can come back in a year or two, resample the soil, see if that diversity has increased, changed, etc. So it's a good way to quantify what you're doing and actually know that you're actually doing something of benefit. And then, of course, see how it impacts the plants. You're working with local cultures. And along the way, these fungi do remediative efforts. So they're not just helping plants and the soil, as I said, but many of these soil fungi also break down chemicals. They sequester heavy metals. They bind up and lock up heavy metals and make them so they don't affect plants and animals. All these remediation efforts and abilities that historically or more commonly people just think about the macro mushrooms doing. But actually many fungi do this. Many of these soil fungi that we never see can break down all kinds of chemicals. Again, this just speaks to the chemical mastery of the fungal queendom as a whole. So when we can cultivate them, bring them in, there's even soil molds that don't even form these symbioses. They're just in the soil doing other stuff, and they break down chemicals. And we know molds grow pretty easily on all of our fruit and bread and stuff. We can grow them incredibly easy. And these are beneficial molds, and put them back in the soil and they'll regenerate. They'll actually support plant health. There's a lot of research with some of these fungi. So again, just broadening beyond the mushrooms that are a good inroad to the vast world of fungi, which is so much vaster.
And also one of my dreams is to co-create a bioregional spore and culture bank amid the old growth redwood forest and other temperate rainforest regions. And I'm wondering if you think it's critical that we clone rare genotypes of fungi from ancient forests as part of the process of preserving biodiversity. How could this be done, and what equipment is needed for an amateur to do this?
Well, I think the spore and culture bank concept is a natural outgrowth of the seed saving movement, which is a contemporary outgrowth of a very ancient practice, around the world. But as far as I've come across, people weren't historically saving mushroom spores intentionally. More cryptically with some fermenting practices, they would put the stick into the beer brew and get the stick infused with the yeast, but they didn't know what they were doing. They thought it was magic when they used it the next time. Today I think that has many obvious implications. It's not a bad thing. It doesn't hurt to do it. It's easy to take a spore print as the basic step. So if anybody wants to do something in this effort, to take a spore print, you take the mushroom, whatever the mushroom is. Any type of mushroom produces spores. Some produce a lot more than others, so you're going to get more spores than others. But take the mushroom cap, or wherever its spore-bearing surface is. Some mushrooms, they produce spores across their entire exterior. Some, it's only underneath the cap, like the shiitake or something that we're more familiar with. Regardless, figure out where the spores are being produced and just set that mushroom onto a piece of glass or onto a piece of clean aluminum foil. Ideally, you prop it up with a couple of toothpicks just so it doesn't get a bunch of moisture onto its own spores as they drop over the next 4 to 24 hours. And after that period, just move the mushroom, wrap up that foil, just fold it onto itself, make a little envelope, or if it's a piece of glass, say like a microscope slide, make a sandwich with another glass piece and tape it. And as long as those spores stay pretty dry, in theory, they can be viable for years, if not decades, if not potentially millennia. We've only looked at it for 100 or 200 years, but certainly 100-year-old mushroom spores can be just as viable and come back to life. And this gets into a greater notion that mushrooms came from outer space because their spores can survive, but that's a huge other topic that I don't necessarily get behind, but it's an interesting thought. And then as far as the cultures go, as far as the actual tissue, and that's perhaps what's even more, I don't want to say important, but just more nuanced, because when you get a spore print, again, you're getting this huge genetic diversity, which all come from that parent mushroom, but every spore is theoretically going to be different than that parent as far as its genetic, gene sequence goes. So if you actually clone the mushroom, which is what we do in a lot of cultivation practices, you literally take a little piece of the mycelium and you preserve that, or you expand it later through cultivation. And when you take that little piece of mycelium, it's like cloning a tree or grafting a tree. You're getting a specific genotype that has specific, abilities and results and taste and medicinal properties, etc. And so that's why we cultivate. That's why we clone. We cultivate because we know what we're going to get at the end. We work with spores. You don't know what you're going to get. And to clone, there's a lot of ways to do it, in really lab-based setting. But a really simple way to do it for anybody that doesn't have the money or time is many mushrooms, especially the decomposing mushrooms we find on old wood, will grow pretty well on cardboard for a couple days at least. So you soak cardboard, strip all the tape off, and try to have as little ink as possible. Soak it in water for half an hour to an hour. Get it really saturated, let it drip off. Go out into the woods when you're hiking or camping. Bring that cardboard into some Tupperware so it doesn't dry out. And when you find a mushroom you want to preserve, take the mushroom, rip it up, throw it into the cardboard, and roll that cardboard. So this is ideally corrugated cardboard, so we want the airflow in there. We don't want just flat sheets because it'll suffocate. Kind of roll that up, get it in contact, and if all goes well, in the next couple of days, you'll start to see mycelium coming out of those chunks. Again, the whole thing is just mycelium, so it'll start to revert back to a vegetative growth. And at that point, you can actually dry out that mycelium and hold on to it because, believe it or not, many species, and I've had pretty incredible experiences where the mycelium is totally dried out. You would assume that it's totally dead, but it's essentially in suspended animation and come back perhaps even years later, who knows, maybe decades later, and just rehydrate the mycelium and it'll come back to life. We know that lichens can do this quite well. Lichens are incredibly resilient, and actually, if anything came from outer space, it was probably a lichen and not so much a mushroom. But even as far as cultivation goes, I've brought back dried out things that were baking in the sun for months on my dashboard, and I brought back the mycelium just by simply rehydrating it. This interesting guy, Bowler, one of the mycologists from the early 1900s, kind of a kooky guy. He did a bunch of crazy experiments with mushrooms. One of them was basically putting the fairy ring mushroom into a vacuum tube and cutting it off from any input, any air, anything for like 40 years, and then brought it out, and it immediately came back to life and started producing spores. So, you never know what you're going to get. Those are some really simple means of spore print, and just dry out the mycelium on cardboard, and it can get more detailed from there. There's better practices, more assured practices, I should say, that are more consistent. But those are nice intros for the beginner.
I got a little giggle thinking about you bringing back your lichens from your dashboard that could have potentially been coming from outer space.
The truth is in the dirt on the ground. The truth is in the dirt on the ground, not in your gilded cage with your rusted spoon. When the ground splits open, it will swallow you. The truth is in the dirt on the ground. The truth is in the dirt on the ground, not in your poison tongue or your acid tears. Let the truth howl in your ears. Here she comes, it's killing time. Flames are burning behind her eyes. Here she comes, it's killing time. Flames are burning behind her eyes. The truth is in the dirt on the ground. The truth is in the dirt on the ground, not in your claws drawn out, scratched till it bleeds. Knocked down, dead gets what she needs. The truth is in the dirt on the ground. The truth is in the dirt on the ground.
We kind of touched on myco-remediation for a moment, and something I wanted to go back to was plastics, considering that they are the number one form of pollution created by humans. And many plastics are reported to take thousands of years to break down. Can you help us understand how fungi could be trained to eat plastic, and how could this process tangibly work on a large scale?
So, fungi being these incredible chemical masters, there's a fungus that can eat anything that humans have ever produced. When all said and done, fungi will destroy everything that humans have ever created, and they will create new life in the place of our human structures and all these things. So anybody that's worried about the world will persist no matter what, and fungi will help that process. So despite the fact that plastic is filling the oceans and the landfills, there are fungi that can break it down, but we are not intentionally introducing those fungi appropriately to speed up that natural ability. Since plastic was created 100 years ago, there's all kinds of plastics. I don't know, dozens, hundreds of times scientists around the world have taken these different types of plastics. They bury a piece in the ground. A year or two later, dig it up and see what happened. And sure enough, every time the plastic is thinner, weaker, discolored, it's being broken down. And it's usually the fungi that are responsible for that. And it's usually a complex of species or assembly or even successional waves of species, as naturally happens with everything else. Different things eat different components. So we know that fungi can break down plastic, but that's pretty much where all that research is ended. They bury a piece, dig it up, test the species on it, say, okay, it's happening. Why hasn't it turned into a large-scale practice to actually deal with this significant problem that we're creating as humans? I don't know. But there's a ton of potential there, and it really just gets back to basic cultivation concepts and really getting the best species for the job. So a great anecdote is some Yale students were growing endophytic fungi from the Amazon. These are fungi that live inside of plants that I've been speaking of, studying these symbiotic, kind of cryptic, hidden fungi that you would never think have encountered a human industrial pollutant deep in the jungle. They're growing these fungi on petri dishes, and one day they notice that the fungus is breaking down the petri dish, the plastic petri dish. So then they grow that mycelium in a liquid form that I've sort of mentioned. They introduced the plastic in a liquid form just to make things simple on themselves. But then they did a bunch of tests, and they were able to show that this endophytic plant-inhabiting fungus was able to eat this one type of plastic as its only carbon source with or without oxygen. So essentially fermenting it inside of a landfill is this idea, like deep in the landfill, it could be eating these types of plastic. So that's just one species that they found randomly. And that made headlines in the fungal mycological world. It's pretty big news, pretty amazing discovery. But nothing's happened with it as far as I know. And who knows how many other endophytes or all these other cryptic fungi, and some of the fungi live in really extreme environments. What abilities do they have? We have no idea how many endophytic species there are in the world. It's one of the least studied ecological niches of fungi, even though it's one of the most ubiquitous, let alone their remediative potential, their plastic eating potential. So we might first think about, okay, the mushrooms, because that's where a lot of the remediation research has gone into, breaking down petroleum products and other nasty chemicals humans produce, PCBs, all these things we don't like. Many mushrooms, many wood-decomposing mushrooms especially, have been thoroughly researched for the last 50 years to show definitively that many mushrooms can break this stuff down, no problem, depending on the species, depending on the chemical. Not as much the same with plastics, and I guess what I'm getting at is we also need to start thinking about beyond the mushrooms. When you think about the whole fungal kingdom, it could be some Sonoran desert fungus that is kick butt at breaking down, plastic number 5 or whatever. So, and that's the type of stuff that anybody could mess around with at home. So a great anecdote is Tom Volk, a mycological professor in Wisconsin, one of the universities there, worked with a grad student trying to figure out how to get a fungus to break down bakelite. Bakelite was one of the first basic types of plastic. It's basically made from phenol and formaldehyde. So it's pretty different than today, but it's technically considered a type of plastic. They tried 84 different experiments to get the fungus to eat the plastic, and the one that worked was when they just straight stuck a chunk of the plastic into the mycelial network. And then the fungus just started breaking it down, dissolving it. They were able to test that, and they actually got a $64,000 grant because of, the photos they were able to get of this working. So the money's out there because there's significant implications, but it just gets into people needing to know about fungi, need to know how to cultivate them, need to kind of wrap your head a little bit around the chemistry, all of which I detail and try to simplify or at least make accessible in the book, not watering it down. As I say, in many ways, to do good cultivation remediation, you're best served to understand fungal biology and ecology, because that's what you're trying to mimic. So many of these implications beyond plastic remediation, all the other remediation, many people, everybody could do to some degree. Smaller, large scale kinds of experiments have yet to be done. But again, I think people just assume that other people are doing it. We also are busy, in our lives. And then the lack of knowledge is, I think, the biggest hurdle. So the plastics, it's one of the things I've looked at for a long time. I wrote a paper a bunch of years ago with some friends looking at this, did a big research project. I touch on it in the book briefly. Basically, the state of affairs is that we know fungi can do that. Nobody's really dedicating it to it, even though it's a global issue.
You mentioned the endophytic species living in the absence of oxygen. So I'm wondering, with the massive plastic islands in the ocean, could this fungus, or perhaps marine fungus, be put to work there?
Well, as far as I've seen, nobody's really looking at that. The issue with the gyres, as they call them, in the middle of the oceans, is it's not just some big island of plastic water bottles or something like that, which is what I initially imagined when I was thinking about these things. The problem is that it's the UV from the sun and the waves are breaking down these plastics into tiny, almost microscopic particles. So it's becoming this thick plastic soup that's up to 30 feet deep. And plankton's eating it, and it's going up the food chain, and obviously there's a lot of issues there. And so when you get really deep, 30 feet down into the ocean, it's really cold. There's not a lot of dissolved oxygen. So it could be hard for a fungus to grow. Of course, fish are living all the way at the bottom of the ocean, and we actually don't even know how to explain that, which is a separate topic. But as far as I've read, I've wondered, I'm sure that there's fungi involved there. I'm sure that if they tested that plastic, there's probably some sort of fungal component that is common or more abundant than others. There's probably a whole bunch of species, because there are yeasts and all kinds of fungi, microfungi, in the oceans. They actually think that a recent paper from April postulated, based on what we currently know, that the entire ocean floor could very well be permeated by fungal mycelium, which is the ocean floor is the largest habitat in the world. So they're literally at the bottom of the ocean, at the bottom of the entire food chain, because that's where everything comes. And they're the ones churning the nutrients down there, bringing all the dead fish that fall down. It's the fungus that's eating it, which is what they do everywhere else, and then they're recreating life. So there are ocean fungi, marine fungi, again, one of these huge topics. Planets 70% water, 70% oceans. We hardly know anything about the fungal component of the oceans, and especially the ones that are able to eat the plastic there or survive in chemical plumes or something like that. Tons of research. So many of these topics. It's each of these questions. It's like a PhD. It's 20 PhDs waiting to happen if people kind of dedicate. It doesn't have to be a true PhD, but it's a huge thing that nobody's looking at. Something I'm totally compelled by, but I only have so many hours in the day myself as well.
It's really enticing to hear a little bit of this information and get the ignition for us individually to go deeper into these thought processes and experiments. And another thing that the mainstream media and politicians deny and don't talk about is that the drought is the logical outcome of the misuse of land. And, there's all these deep patterns and dots to be connected on this topic as well. For example, we only have 10% of the wood debris left in the forest compared to a few hundred years ago. And considering mycelium is 90% water and holds water in the soil, a major byproduct of mycelium decomposing wood is water. And so decomposition is like drip irrigation for the other organisms of the forest. Also, the great forest drew in the rains and stored water. Do you think it's possible to reintroduce or boost fungi in ecosystems to help retain and replenish groundwater, and ultimately to alleviate droughts that are inevitably going to get worse as climate changes?
Well, I strongly lean away from ever making hyperboles about, or stating hyperboles about fungi, or putting all of the world's problems onto their backs, because that's how they can be presented. And I think for some people that I've met, myself included, many years ago, when you don't fully understand them and you hear about these really incredible things, it's easy to want to put all of our hope into this, mystery, and to say, well, if they can fix this, then they must be able to fix this, and hey, they can fix every problem in the world, right? And we see this in many ways, that fungi, I think, are abused and misrepresented, and it's something that I sometimes have to burst bubbles around. be more, I think, respectful of the fungi. And again, their limits, and also take some ownership about what we are contributing as individuals, and what else needs to be done more systemically. So, I mean, I think you sort of answered your question that these fungi are clearly an integral component. They provide all these answers. They provide all these benefits to the larger system. So they're not doing everything. I mean, plants do stuff, animals do stuff, insects do stuff, bacteria do stuff. All these functions are important to the whole ecological web. Fungi are essential to that. They're keystone species, but they can't do it alone. So we always want to bring them in. We always want to consider them more. We want to advocate for fungi. Actually, a friend of mine, Juliana Furci in Chile, she runs a nonprofit NGO called the Fungi Foundation, and she basically travels the world trying to get government agencies to implement fungal analyses into their ecological surveys, because most places are just looking at the flora and fauna, but not looking at the fungi. I mean, sort of restating stuff I've already said, if we're going to try to do any restoration and we're trying to protect an ecological habitat, we want to, of course, protect the fungal component, advocate for it, strengthen it, bring in perhaps species that have been eradicated through human interventions, reproduce and diversify and expand the species that are there. You know, and with any of these restoration efforts, we always want to be slow and use simple solutions. We want to be like mycelium and assess really thoroughly, and also see what the environment is doing. That's a pretty basic permaculture concept. And we always want to learn from the fungi and see what they're doing, see what species have come in, and never assume that we as humans, with our very limited awareness, have all the answers to nature's 3.8 billion years of knowledge acquisition that we call evolution. So, yeah, like I said, I think you answered your question. I mean, so much of what I do is just trying to say they're not the silver bullet. You know, every hypha can seem like one, but they're an incredible tool that most people just don't know is in the hardware store. And they fit right in with all the other tools in the toolbox. We just got to make space for them.
Reality is a word some people don't understand. They spend their whole life living on make-believe plans. They think life is a fairy tale full of joy and ecstasy. But wake up, oh, oh, wake up and face reality. Reality is life, and sometimes it's a test. There will be hard problems, but you do your very best. So stop fooling yourself, open your eyes and see. But wake up, oh, wake up and face reality. Reality is life filled with tears. Reality is sorrows and tears. Reality is yours for the asking. Reality is life everlasting. Reality is heaven if we only open our eyes. It can be more beautiful in the moonlit skies. Reality is ours, waiting for you and me. So wake up, oh, wake up and face reality. Wake up, wake up, wake up, oh, wake up now. Oh, baby, won't you stay to wake up now?
Well, thank you for bringing that full circle back to our responsibility as humans and not putting all of the pressure on the fungi, but realizing that we actually have to change the way we live and really have a reciprocal relationship with nature and not just expect that there is a silver bullet or a magic answer out there. I think that's really important. while we're talking about all of these really exciting tools and all of these possibilities when we ally with the natural world. And so in that spirit, I'm wondering if you can suggest any projects or calls to action or other ways radical mycology could start transforming our communities right now.
Well, I mean, the great thing about mycology is it does touch everything, and as I've been saying, and so the ways to apply it are almost innumerable. I mean, you can get as large or small as you have time and capacity for. It's the knowledge gap, it's the access to information that's the biggest hurdle. I think initially the biggest hurdle, then it gets into resources and just funds and time and stuff. But even then, once you have the knowledge, once you have the awareness, some of these skills are pretty simple, whether it's cultivating mushrooms for your own food, recycling some newspaper waste in your neighborhood, kind of like the reverse newspaper boys, how my friend calls it. Instead of giving out newspapers, pick them back up and turn it into food and medicine. It's a pretty cool application of these concepts. If you want to get top down, if you're that type of person, you want to work with your local bureaucracy, whatever. I'm not going to harsh that. If you're trying to mitigate waste streams and you're trying to reduce pollution, because fungi are the loop closers, they're nature's grand recyclers, and we can ally with them and bring them into our human systems to close our loops a lot tighter. And then it gets into, local medicine, cultivating the local natural medicine from your environment is better than importing it. And that clearly aligns with a lot of our herbal movements. There's a whole radical herbalism movement that's a bit underground, and a big part of that is advocating for free natural medicine for folks. In Olympia, where I used to live, there's a free herbal medicine clinic. It's an incredible community resource of volunteers that are passionate about healing those around them with natural medicines. Fungi obviously align right with that. They're natural medicine, and we can bring them into our discussions around these day-to-day needs. And then if we have more time, resources, tons of potential with the remediation stuff. As I've been saying, there's so many ways to integrate fungi into a damaged or disturbed habitat to try to assist the natural process that is already occurring to come back from those shocks, whether it's a natural or human-caused shock. Again, given some geological timescale, 5 years or 5,000, a habitat that's been incredibly impacted will recover. All the radiation we've dropped will go away in a bunch of thousands of years. It'll go away at some point. It's just going to take a long time. So nature has its ways, but we can help with our knowledge, our hands, our thumbs. We can actually speed up that process and work with it. So, and that's what I detail in the book, the remediation chapter. So even there's small little things. So a couple of years ago, I made a video about how you can grow mushrooms on your old cigarette butts. Cigarette butts are like a sponge full of the nasty chemicals from the cigarette. Mushrooms can eat those chemicals, and instead of it leaching out of the cigarette butt, in the environment, the fungus can break it down, and that way it's much less toxic. And you can do that on your back porch or on your front porch, wherever you smoke. And you can scale that up. One of the things I talked about in that video, and something I still advocate for, hopefully one day we'll get the time to pursue, is actually turn that into a citywide practice where you incentivize a refund policy, kind of like a bottle return. For every pound of cigarette butts, you get X amount of money. And in return, the city or the organization will grow mushrooms on that, break down those chemicals, and you can make functional objects out of that mycelium. Mycelium is like the new plastic. It is going to be the plastic of the future. There's a pretty good Vice video talking about this. Came out about a year ago, called Fungus, the Plastic of the Future. Mycelium is one of the greatest building materials, natural building materials. We can grow it in any shape. It's buoyant, incredibly flame retardant, impact resistant, obviously biodegradable, but incredibly rot resistant when it's dry. It's lightweight, so many things. Time's just waiting for people to know how to cultivate it into a shape, and we can make pretty much anything that's currently made out of styrofoam, cork, wood, etc. We can make it out of mycelium in the not too distant. We can make it today, but we're not up to speed yet. Our planes, our cars, whatever. If we want to stay civilized, we can do it a lot better, certainly. And there's a lot of ways to do it. We don't have to get rid of all the comforts we've developed. So that, obviously that's a different direction that maybe not everybody listening will want to go in. If you want just immediate solutions, fungi provides so many. Creating better food and medicine is the initial ones that most people jump on or can kind of get behind easiest, and it's most accessible. But cultivating these soil fungal cultures, creating the spore and culture banks like you're talking about, advocating for fungi in a variety of forms, destigmatizing them, and then from there having fun and learning with others. I've always advocated for forming radical mycology groups, or whatever you want to call them. I don't really care if you use that term. Just people getting together around this really niche topic that is obscure and kind of hard to access, learning together, and then figuring out what's most immediate for your needs. I mean, that gets into every hypo tip. You know what you need for your environment. You know what you need for your home and for your life, and if you work in your community, what the community needs. Mycelium, fungi, mycology can all benefit and support any of our efforts to whatever capacity we can give them.
Wow, Peter. Well, thank you so much for, just an incredible analysis of the fungal queendom. So many different benefits and uses and ways to ally, and I am deeply moved by your relationship with the natural world and how you have chosen to stand with the mushrooms and the mycelium and the world around you and making it accessible for wider groups of people. It's an incredible life's purpose, and I'm so grateful for all that you do. And I know a lot of the audience members will feel so similar and will want to learn from you and be a part of what you're doing. So if you could just mention where people could find you online, where you'll be teaching next, where to get this incredible resource, the Radical Mycology book. Yeah, just where we can keep in contact with you.
Yeah, thank you. I really appreciate such great questions. I usually don't get such probing things, and these are really where my mind goes with these topics. But I'm always happy to meet people where they're at, but I love your show for this reason. The book is available through the publisher. It's actually my own publishing company, self-publish it, working with a friend or two to help, but you can support that. And the website is CHTHAEUS, Catheus.com. Catheus is a word I made up. It means both underground and the highest state of matter before it becomes ether. And so it's a combination of those two, which I think fungi embody. So you can buy the book there. If you go to radicalmycology.com, you can check out some free resources we have on there. We're always hoping to update the website, never enough time, but we're working on that. And actually, we are making headway to revitalize the site sooner than later. But there's some good stuff there now. And you can also, on the Radical Mycology site, check out the book tour going for 3 months around the whole country. And as a part of that, I'll be doing in about 11 different cities weekend-long cultivation courses, so more in-depth courses going into a lot of the cultivation practices, remediation stuff a little bit, medicine a bit. So the descriptions for those courses are at the website. And then in between those courses, the day events are all free readings and presentations. I'm going to be trying to make my vehicle into like a fungal roadshow. So I'll be trying to bring a bunch of stuff and make it interesting. So check that out. I'm coming to like 40 different cities. And then, as I mentioned, in October in New York, we're having the 4th Radical Mycology Convergence. And that's at radicalmycologyconvergence.com. So hopefully folks can make it out to that. Should be really amazing. We're leveling up in a bunch of ways and super pumped for all the new stuff we're doing this year. And then otherwise, you can get on the email lists on both those sites, stay up to date. Later this year, and especially next year, I'm going to be doing a lot of new courses based on the stuff in the book. So really expanding beyond cultivation. So folks can definitely stay tuned for that. And otherwise, I'm around. So just send me an email and say hi.
Drifting on the wind through the mountains like a river.
Hello again, this is March Young. Thank you so much to our guest Peter McCOY and our host Ayana Young. The music today, Silence Returns, a 1971 track by Bo on John Peel's Dandelion Records. Then Karen Elson, The Truth is in the Dirt, and Al Robinson with Wake Up. Our theme song is Like a River by Kate Wolf. Thank you again for joining us.
Sweet smell of pines, tall western cedar, drifting on the wind through the mountains like a river.